See it fine.
Looks
So
good.
we're really delighted to be able to present Autism BrainNet to the Spark community. So we appreciate this opportunity. We're going to do a tag team presentation today. This is the outline of the presentation. I'll do a little bit of an introduction about Autism BrainNet. We will then have period of looking at very sort of overview of brain development. We'll talk about why brain donation is important for autism research, what researchers are learning about autism from postmortem brain studies. And then Carolyn will talk about topics like who can become a brain donor and what is the donation process. And then at the end, we'll also talk about what is the request process for clinicians who may want to get some of this resource. So what is Autism BrainNet? Autism BrainNet, first, our mission is to work with scientists around the globe to advance research into the causes of autism and related neurodevelopmental disorders. Autism BrainNet is a program of the Simons Foundation and was established in 2014. Our goal is to build an essential resource of postmortem brain tissue for research into the causes of autism and related neurodevelopmental conditions. And since our inception, we have acquired nearly 400 postmortem brains. We're really a team effort. This diagram sort of illustrates first that we have 3 collection sites. Let's see if I can get that going. Yeah, so we have a collection site in Sacramento here at the Mind Institute. We have one at UT Southwestern in Dallas, Texas, and we have a third collection site at the Mass General Hospital in Boston. We have a specialized collection in New York, and recently we've also established a collection site at the Douglas Bell Canada Brain Bank in Montreal, Canada. In addition so at each of those sites, we have a team that can oversee the collection and preparation of brain tissue and storage for later scientific investigations. Beyond that, we have a team of administrators, including Dr. Marta Benedetti, who's at the Simons Foundation and is a co-director of Autism BrainNet. David Layden, who handles much of the financial interactions of Autism BrainNet. And then an outreach team, Lillian Acosta-Sanchez and CC McBride, who reach out to organ procurement organizations and others around the country, trying to make them aware of the need for postmortem brain donations. And of course, both Carolyn and Nicole Coman provide the clinical interface between Autism BrainNet and donor families. So I'm going to turn it over to Carolyn now, who will talk about our partners and then about our collection.
And thank you, David, very much. And so our team really does extend beyond the folks that David just described to you. Our team extends really to you as the Spark community and a multitude of patient advocacy groups. We, of course, also work closely with Simon Searchlight, various medical examiners, organ procurement organizations, tissue banks, and other folks you see listed here below. We rely very heavily on our community partners to understand the importance of brain donation for research, and of course to understand the process by which brain donation occurs so that we can collectively support and advocate for our families in their time of need. The last point that I want to make here on this slide is that this isn't an exhaustive list of the patient advocacy groups that we work with. You can certainly have a rare genetic condition, not be part of one of the groups listed, and be a donor to Autism BrainNet. And in fact, we'll talk a little bit more about that when we get to our inclusion-exclusion criteria down the road. And so, as David mentioned earlier, we have nearly 400 donors to our program at this point. And moving to the next slide will give you a sense of who are the donors already to our program. You can see that we have just over 160 control or typically developing donors, and I always want to kind of put quotes around that. We have 48 donors who have a neurodevelopmental or rare genetic condition without a diagnosis of autism. And then we have 155 donors with a confirmed autism diagnosis. And it's important to note about that population of folks that they may also have a co-occurring genetic condition that really is the root or the etiology of their autism. And here on this slide, just looking at our collection and from a different perspective, you'll see that we have sort of a disproportionate number of control donors in that 41 to 50-year age range. And really, we're lacking control donors for our youngest and our oldest donors. And we'll talk a little bit more about that too when we get to our inclusion-exclusion criteria. And in terms of our donations, looking at it from a gender perspective, you'll see that for our donors with a diagnosis of autism, we have almost exactly 4 times as many males as females, which is really consistent with what we know about autism as a diagnosis. Interestingly, our folks with rare genetic conditions that do not have autism, it's an equivalent number of male and female or, male and female donors that age.
Okay, so having given a brief summary of our collection, we wanted to talk a little bit about brain development in general and sort of make the case for why it's important to study the postmortem human brain. So first of all, I think just taking a step back and thinking how complicated it is, the process of human brain development. So the human brain has on the order of 100 billion neurons. And those neurons make different number of connections with each other, and they make up to 150 trillion of these connections, or what called synapses. 150 trillion is the number of stars in the Milky Way galaxy. So it's a very large number of connections in each of our brains. And this all happens during a protracted developmental period, but is very rapid during prenatal life. So the rate of neuron production in the human brain is approximately 215,000 neurons per minute on average during the course of pregnancy. That equates to about 15 million neurons being produced every hour during pregnancy. In reality, the number of neurons that we're going to have in our brains are pretty much accumulated by the end of the second trimester. There are some special places in the brain, like the hippocampus and the olfactory bulbs, where there's ongoing neuron development throughout life. But basically, we have our complement of neurons by the end of the second trimester. But that doesn't mean that brain development stops by the end of the second trimester. In fact, you can see this image on the left-hand side of the screen. This is now at term at 9 months. And at that point, the brain is only about 25% of the size that it will ultimately be in the adult. It reaches about 95% of its adult size by age 6. And so what's going on during that period? Well, this is an image of each of these black dots are a cell body of a neuron. And what you can see in a 1-month-old child, the black dots are really densely aggregated together. And then what happens over time in the 6-month-old child and the 24-month-old child is the dots get bigger, so the cell bodies actually enlarge. But most importantly, the spaces between the cell bodies are also expanding. And what fills those spaces are called dendrites. And these aren't all the neurons. If there was all the neurons and all the dendrites were illustrated, it would just be a black gamish. But we're seeing a small percentage, about 1% of the neurons. And you can see the dendrites look like trees. And it's the dendrites where the vast majority of the connections from one neuron to the other are made. And these dendrites continue to grow up until uh, both 6 years and a little bit beyond, in some places much beyond that. So that's a very active process. This process of the brain maturing over time, there are techniques to be able to look at that in the living child. So these are images from using magnetic resonance imaging work that we do at the Mind Institute at UC Davis. We do longitudinal studies of the development of the brain of both autistic children and neurotypical children to see if we can detect any differences. And here you can see that between 1 week and 2 years, there's very substantial development of the brain, and that continues on to 10 years. Now, the thing about magnetic resonance imaging is it's a wonderful technique to give sort of a global overview. I oftentimes think of it as like looking down at the earth when you're riding an airplane and you have the window seat and you're looking down and you can see Lake Tahoe or you can see Lake Michigan. But what you can't see is the kayaks that are kayaking on Lake Michigan or Lake Tahoe. And so in terms of the brain, you can actually see the individual neurons or the molecules that are within the neurons using magnetic resonance imaging. For that, you have to do postmortem brain analysis. Now, the question is, what kinds of things do we see with MRI? And there are some findings that are actually very intriguing. So, for example, this is again work from our group at the Mind Institute. What we found is that about 15% of boys have a brain that is actually too large for their body. Something has been altered in brain development. And so what you see here is that these are 2.5-year-old children, and it's a surface rendering of their brain from MRIs. And this is what a typical child's brain looks like. It's about 1,000 cubic centimeters. Here's an autistic child uh, that has a brain about the same size, but almost 1,000 cubic centimeters. But here's one of these children that has a brain that's actually too large, and it's 20% larger than the children, the autistic children with the normal brain size and the typically developing children. Now, as it turns out, um, children who have this large brain form of autism, their cognitive status is actually more challenged than the individuals with autism who have normal brain size. So not only do they have less gains in IQ during childhood development, they have less language function, and they seem not to be able to respond as well to behavioral interventions. So their prognosis is worse than children that have autism and a normal brain size. So we can tell that the kids have a larger brain, but we don't know why. And again, being able to determine why the brains are enlarged, what might be causing this poorer prognosis for this 15%. And I should have said that this is actually more common in boys than in girls. 15% of boys have this enlarged brain version of autism. It's only about 3 or 4% of girls for reasons that, again, we don't quite understand at this point in time. But why is the brain larger? We don't know. And this is one of the things that we feel is important to find out, but can only be found out by studying postmortem human brains. Now, you might ask, why can't you do this in animals? And there's a variety of reasons, but here's some illustrations just to give you a highlight. This is a mouse brain, and this is a human brain at the same scale. I already mentioned that the human brain has about 100 billion neurons. The mouse brain only has 71 million neurons. And what you can see is that in the human brain, the huge expansion of the brain is in this region here and in this region here. This is the frontal lobe, this is the temporal lobe. And these are regions that have shown the greatest expansion in our evolution. And they are actually the regions that we think are most germane to understanding autism. And as it turns out, there's large portions of these regions that you can't even find in the mouse brain. So the mouse brain isn't just like a mini human brain, it's different. And many of the regions of the human brain that we think are going to be important for understanding autism simply don't exist in the mouse brain. And here again, this is just taking a section through the brain, but you can see here's the mouse brain here. When you compare that with the human brain is far more complex, has many more connections. It even has cell types that aren't found in the mouse brain. And so, again, in order to understand a human disorder like autism, you have to study the postmortem human brain. Now, we come at this from a long history of people studying the human brain to understand neurological disease. Alois Alzheimer's published a paper in 1907 where he found that a woman who had presenile dementia had peculiar characteristics of neurons. He called them neurofibrillary tangles in her brain when she came to autopsy. Subsequently, people have studied this now for more than 100 years and found that there are both what are called senile plaques and tangles, and these are the hallmark features of Alzheimer's disease. And many of the therapeutic interventions that are now being tried are based on the fact that we have an understanding of what's causing these plaques and tangles, and the therapies and preventative measures are trying to eliminate these plaques and tangles or prevent them from ever forming. So with autism, unfortunately, we're sort of in the early stages of understanding changes in the autistic brain. So autistic neuropathological studies only started in the 1980s, not in the early 1900s. So far, we haven't found a consistent sign like plaques or tangles in the autistic brain. And in part, that may be because autism is so heterogeneous, but we haven't seen a smoking gun yet in the autistic brain. But there are things that we've seen studying the postmortem human brain that is giving us insights into what's going on, what kind of alterations might be there in the autistic brain. So before I tell you about some of the findings that have come out of postmortem research, I just want to emphasize that the reason that we think it's important to study the autistic postmortem brain is because the brain is the primary organ affected in autism. And if we're going to understand it deeply, we have to understand the alterations that are taking place. The postmortem brain research can lead to diagnostic biomarkers, more importantly, potentially therapeutic targets. And ultimately, the goal of all of this work is to improve the quality of life of individuals with autism. Again, there is a history of where, by understanding the changes in Alzheimer's, Huntington's, and Parkinson's disease, for example, we are able to now develop therapeutic targets to help individuals with these disorders and diseases. And I want to also make the point, I think Carolyn will also make it again later, but that brain donation is not part of organ donation registries. So if you have an indication on your license, for example, that you want to be an organ donor, that doesn't let anybody know that you want to donate your brain to research. Now, organ donation, of course, is life-saving for helping people that may have a failing organ for transplant. That doesn't happen in brain donation. Brains aren't obviously donated to other individuals, but uh, brains are incredibly important, as I've been trying to say, for understanding the causes and the trajectories of disorders like autism. So you have to let people know that you want to be a brain donor. And you have to think about it ahead of time, as we'll talk about a little bit later. So let me just tell you a couple of examples of research based on postmortem brain donations that have already made a contribution to our understanding of what autism is. So there is a theory that that a lot of the symptoms of autism. So let me take a step back before I say that. So in the brain, I just been talking about neurons, but obviously there's different kinds of neurons. Basically, there you can think of it as that there's excitatory neurons that stimulate the next neuron when they communicate, and there are inhibitory neurons that actually depress the next neuron when they get stimulated. So when you have the excitatory and the inhibitory neurons, they keep each other in check and they maintain a homeostasis of activity in the brain. If you have too little of the inhibitory factors, for example, that leads to epilepsy. Well, there's been a notion for a number of years now that in autism, the sensory sensitivities and some of the other features like the epilepsy and autism might actually be due to an imbalance between the excitatory and the inhibitory factors or influences in the brain. And what this group, led by Veronica Martinez-Cedeno, has found, she found, first of all, their team found that there's a group of inhibitory neurons in the cortex, particularly in the prefrontal cortex, called chandelier cells. And in a first paper published several years ago, she found that in autistic brains, there's a smaller number of chandelier cells than in typically developing brains. But there were some issues about how she identified the chandelier cells, and she wanted to replicate that. So what she did was, in this study, looked at not the cells themselves, but the connections they make, the synapses. And the synapses for chandelier cells are called cartridges. And so what they did was they took sections through the autistic brain and the control brain. And in this picture, the control brain images are on this side, and here's the autistic brain images on this side. And everywhere you see a little hash mark, that's where they saw one of these connections of a chandelier cell. And these numbers, BA9, 46, and 47, just indicate different regions of the frontal lobe. And what you can see, I think, is that in the autistic brain on this side, there's fewer of these hash marks. There were fewer of the connections. And this is just where they quantify it. So this is the number of connections or cartridges in the control cortex compared to the autistic cortex in this region, and this region. In all three regions, they found that there were a few of these inhibitory connections, indicating that the prefrontal cortex in these patients during their lives may have been hyperexcitable. And in fact, again, may have led to some of the symptomatology that they had during their lives. So again, this is a study that clearly needs to be replicated, more needs to be done, but we're starting to understand from these kinds of studies what's the differences in neuronal combinations in the autistic brain that might lead to some of the symptomatology. There's another, now I'm going to switch to genetics. There's another finding recently that's really, really new, and it's only come about through the ability to do next generation sequencing of the DNA in individual cells of the body. And what people have found is that there's two kinds of mutations. If there's a mutation that comes from the egg, the mom's egg, or the father's sperm, it then affects every cell in the body of the baby, and that's called a germline mutation. And you can just see this blue baby, all the cells have been affected. But there are also mutations that take place in individual cells as they divide within the tissue of interest. And when those mutations, when this blue cell divides, it causes a little focus of mutated cells, just indicated here by the foot that it has a mutation on this little baby. And I wanted to go back to earlier slide where I said that, remember, neurons are developing at 250,000 per minute. And so when you have that many neurons generating, of course, what they have to do is they have to replicate their DNA. They have to divide, again, replicate their DNA and divide. And through that process, there's errors. It causes little focal mutations. And what Chris Walsh at Children's Hospital and his colleagues have been showing is that when you analyze the human brain for the presence of these mutations, you find them. You find somatic mutations within the brain, and they vary. So each one of these brains actually shows the distribution of a specific mutation. So it turns out that some of these mutations can be very widespread and affect all the neurons of the brain, but maybe not other organs of the body. Whereas there are mutations that arise really focally and only affect a small area of the cortex. And again, you would never know this if you were studying a mouse brain, because many of the areas that have these mutations are, again, not in the mouse brain. And what Dr. Walsh and his team has found, and it's just this highlighted sentence down below, is that ASD brains show an excess of somatic mutations in neural enhancer sequences compared with controls, suggesting that mosaic enhancer mutations may contribute to ASD risk. So I couldn't even begin to start to explain to you what that sentence means. However, the punchline is that by analyzing both autistic brains and typically developing brains, Dr. Walsh and his team have found that not only are there somatic mutations in the brain, but there are different ones and an increased number in the autistic brain that ultimately might explain some of the symptomatology and the alterations in brain development that I've been talking about. The final example is work that we've been doing where we tried to relate behavioral symptoms in living children to postmortem analyses. And this is a colleague, Connor Kearns, who's been working with us for a number of years. And you see, we published a paper together, Clinically Significant Anxiety in Children with Autism Spectrum Disorder and Varied Intellectual Functioning. Really interesting paper. I think if you're interested in anxiety and autism and how it's distributed and whether you see it as commonly in autistic children that have intellectual dysfunction or not. But the point that I wanted to make is that from this paper is that in these children, were 6 to 11 years old, about 8% of typically developing children had an anxiety diagnosis, whereas nearly 70% of autistic children had an anxiety diagnosis, as well as their autistic symptoms as well. And we've been very, very interested in understanding co-occurring conditions in autism. We have particularly been interested in anxiety because we have an interest in a part of the brain called the amygdala. And the amygdala is this little tiny region here outlined in red. It's very small. It's about the size of your thumb. And the amygdala, it's been known now for quite some time, is the part of the brain that allows us to sense fear. So in this example, the visual system uh, brings in information about this snake, and it goes through different areas in the brain that allow us to get a representation of the snake as a complex object. But it's not until that information gets fed into the amygdala that we start realizing that this is something that's dangerous and that we have to be afraid of. And then it's the amygdala that generates all of the heavy breathing, the sweating, and the changes in heart rate that we know as a fear response, like that. So we have hypothesized that it's something about the amygdala in autism that might be leading to the increased level of anxiety in autistic individuals. So my colleague Cindy Schumann, who is actually one of our node directors for Autism BrainNet, worked with a postdoctoral fellow in the group, Tommy Avino, and they actually studied the cellular organization of the human amygdala in autism and in typically developing individuals. And they are able to microscopically count the number of neurons in different regions of the amygdala. And again, the punchline here is that by studying the human amygdala, we came to a conclusion that I think is unique, and that is that first and foremost, in the human brain, the amygdala is a place where this graph is just showing how many neurons there are in children, and then as you get older and older, and it was ending around 50 years of age in this study. And what you see is the amygdala is a unique area in the sense that there's increasing number of neurons in the amygdala over time. So there's more in the adult than there is actually in the child. In autism, the trajectory is very different. So there's more neurons than typical in the really young children, but then there's a period where the number of neurons decreases so that there's a significantly less number of neurons in the adult. We don't know if this is a degenerative process. We still are trying to figure out what leads to this loss of neurons. But there's evidence coming from animal studies that show that if you have too many neurons in the amygdala, it could increase the level of anxiety and inappropriate fear responses, and that if you increase the level of anxiety, increase the level of stress, ultimately leads to a loss of neurons. And we think that this might be explaining what's going on in autism as well. So there is this scenario where we have this very unique human situation where in autism, there's too many neurons in the amygdala early on, too few neurons in the amygdala in adulthood, and we would have never been able to discover this had we not studied the human brain. So I hope those examples give you a sense of why we think it's important to be studying the postmortem human brain. And I'm going to turn it back over to Carolyn now to let people know how they can contribute and become a brain donor to Autism BrainNet.
David, that was an excellent talk. Thank you so much. I learned a lot. Okay, so we promised a little bit earlier in our presentation that we would come back and talk more specifically about our inclusion criteria, as David said, who can be a donor to Autism BrainNet. And starting with any individual that has a rare genetic condition that confers a high risk of autism or neurodevelopmental disorder can be a donor to Autism BrainNet, whether or not they have a diagnosis of autism. We do include, of course, anyone who also has a diagnosis of autism. But I think a lesser known initiative is that we require, as David really clearly illustrated in his talks, donations from folks who do not have a diagnosis so that we can do these comparative analyses. And you can see that today we are accepting donations from our typically developing or control donors up to age 50. Now, I think that'll change over time as our population ages. We'll need those older control matches. It is very important that our typically developing or control donors do not have any serious neurologic or psychiatric condition that could confound researchers. We do accept donations from folks who have a sudden unexplained death due to epilepsy or a sudden unexplained or unexpected death without epilepsy. And then moving to the next slide, in addition to, so again, for our donors without a genetic or diagnosis of autism, genetic condition or diagnosis of autism, it's important that those typically developing or control donors do not have severe head trauma. So that could be chronic or acute head trauma. Anyone who has experienced cancer treatments that are, with chemotherapy or radiation, particularly that might affect the brain. Again, just restating that we have to exclude folks with a neurologic or serious psychiatric condition, as well as folks with known bloodborne pathogens, hepatitis, HIV, and then, of course, chronic or long-term alcohol or substance abuse, because it does compromise the integrity of the brain. Those are rule outs for us as well. Now, this is a part that is very important for everyone to be familiar with and in tune with. Starting with the very top of the screen, this is our 24/7 helpline. So when someone is in a situation where death is imminent or has occurred, it is very important to call that 877-333-0999 telephone number. And this is because, as David said earlier, brain donation is very separate from organ and tissue donation. It does require specific initiative on the part of the family to make it happen. So you call that number and we'll help you through the rest of the process. After you have connected with us on phone, we'll talk you through the informed consent process. We will work to coordinate the recovery, tissue recovery, and then, of course, to safely transport this really important donation to one of the collection sites that David identified very early on in our presentation. And then the third step of the process is that Nicole or I will follow up with our family so that we can learn as much as possible about their person and capture as much information as we can so that researchers know exactly who we're talking about and who this person is as they're looking at potentially structural differences in the brain tissue. And then you see there at the bottom a couple of points that I just want to call out. We are often asked if there's any cost to families for donation, and the short answer is no. We're the beneficiaries of the generosity of the Simons Foundation, and they have made a point to make this process cost-free, not just for families actually, but also to make this resource available for researchers as well, which we'll talk a little bit more about. The last part, so you saw the first 3 steps of the donation process. We do also, for families who want to stay in touch with us and stay engaged with the program, we want to continue to honor their person who has become a donor, and we do that through the Donor Family Aftercare program. Involved in this are, regular letters to families. And when I say regular, I don't mean weekly, but annual letters to families. We provide all our families with a special token of remembrance for their person. We offer them the opportunity to put a tribute along our Memories of Hope website, which is really beautiful. If you would like to take a look at it, what some families have done to honor their person. And then, of course, you can opt in to receive quarterly updates from our program. And as I mentioned earlier, we welcome the opportunity to stay in touch with, provide support for our donor families, some of whom we've been in touch with for 20 years or more. All right, so if thinking about donation is something that you want to continue to pursue, we have so many resources available for our families that were developed by our amazing outreach team. So the important piece to know is that, the intent to donate or signing up for our newsletter, those are all non-binding sorts of commitments to the idea of donation. It's, again, very important to call that 877 number when the time comes. Pre-registration is certainly not required for anyone to become a donor to our program. And lastly, for the researchers, which we learned are in the audience, thanks to Dominique's poll at the beginning of the presentation, we want to let our researchers in the audience know how to access tissue and clinical data that is available to them. So you go to Safari-Base, the instructions are here. I'll point out the email address, which is requests@autismbrainnet.org. Any questions that you have about accessing the resources available through Autism BrainNet, you can have answered by emailing that address. And the point that I'd like to make for our researchers here is that our donors as people and are incredibly well characterized through the follow-up interviews that Nicole and I do, but also through whole genome and exome sequencing, through a minimal or research neuropathology report. And we do also, at the time of collecting postmortem brain tissue, we have started taking small dermal biopsies. We do also collect whole blood for DNA, and when parents volunteer, we also collect saliva from them. So those are resources that are available to researchers. If, when you get to Safari-Base, there's something that you're interested in, if it's not easily or readily available, just ask, because chances are that we've collected the information. We just haven't made it flow through to Safari-Base. So please reach out with any information that you're specifically interested in understanding about our donors. And we'd just like to thank you. David, I'll turn it over to you to wrap us up.
Yeah, so thank you, Carolyn. Very nice. And again, a lot of this information that we briefly touched on today can be found on our website, autismbrainnet.org. Again, you can send questions to info@autismbrainnet.org if you have specific questions, or you can call the 877 number and post some questions there. Again, it's important to have the discussion ahead of time. I know this is a really difficult topic, and I often say that each of our donations is bittersweet, because we know that with the donation that we receive that can facilitate science, some family has gone through a very difficult time. It's not at that very difficult time when you should be thinking about making a brain donation. It's really well ahead of that. Either if you're an autistic individual, you can certainly make the decision on your own. You should let family members know that that's your intention. But if you're a family of an autistic individual who would find it difficult to make that informed decision, and you have to make that decision for them, it should be a discussion with the family well in advance. And again, just to bring closure on the idea that the brain tissue is important. It is optimal for research if we receive the tissue within 24 hours. And so it's something that, again, we will help you with if you decide you want to make a donation. But to make that initial decision is something that you should think about ahead of time. And with that, I think we'll go ahead and stop. And we're happy to answer any questions that may have come up. I'm going to stop sharing at this point. Let's see. Stop sharing.
Great. Well, thank you both so much. I think I know I certainly learned a lot, especially about the science side of things, and hopefully this helped answer some folks' initial questions. We do have a nice amount of time for Q&A, so I did want to first start with a couple of more of the science side of things, question-wise that came in, and then we can focus on some of the donation specific questions. Just a
Sure.
couple of some science stuff that
Mm.
came in. So just I think you touched on you had that great slide that showed the comparison of brain sizes from the mouse all the way up to the slice of the human brain. But can you
Mhm.
talk a little bit more about what it is that in post-mortem brain tissue, like what research that allows us to do that we can't do with neuroimaging technology? Maybe just a couple examples or like a high level
Sure.
explanation.
Right. So in the study that I mentioned about the amygdala, for example, we had previously carried out studies where you can outline the amygdala and you know whether it's bigger or smaller, but we don't know whether that's due to the fact that there's more neurons, more connections. There's even another type of cell in the brain called glial cells. And glial cells have been talked about as being important for autism as well. You can't see any of that stuff in an MRI. It's only when you take sections through the amygdala and stain them in different ways that you'd be able to identify and then be able to count and see whether, for example, there's in the amygdala as well, there's evidence that for inflammatory processes there that microglia get activated, they get bigger and fatter and they get angry looking, and that's because they're warding off an infectious process. You wouldn't see that in the MRI at all. Moreover, the kind of stuff that I mentioned that Chris Walsh and his team does at Children's Hospital, there's no way with the MRI to being able to look at genes and the genotype within neurons or even large areas of the brain. So, anything that's at the cellular or molecular level, you actually have to have the human tissue to be able to explore.
Great, thank you very much.
Okay.
And sticking to more of the science here, again, it was another really nice graphic to see the three different brains of the 1.5-year-old
Yeah.
children. And you mentioned so 15% of boys with autism have enlarged brains, but for comparison, do we know the percent of brains of neurotypically developing boys that have larger brains?
Yeah, we use a cutoff of around 3% of neurotypically developing boys would have, that enlarged brain size. But here's the the real interesting fact about that. In general, if you're a neurotypically developing person, the bigger your brain, the better off you are. So there's a correlation, a positive correlation between brain size and IQ. The bigger the brain, the higher the IQ, right? Um, it's not to say that a person with a small brain couldn't have a high IQ, but I mean, there's a correlation. For these kids with autism, the bigger their brain, actually the poorer their IQ and the less language function they have. So the correlation goes in the opposite direction. That would suggest to us that some alteration in the normal brain development has taken place. The brain's bigger, but it's not helping those children out. And we would like to understand what has been different in the is it that too many neurons were produced? Is it, and they don't make the right connections? Or is it one type of neuron that's been produced? Or is it the glial cells that have been produced? We really don't know. That's going to, take, enhanced analysis of postmortem brain studies. Now, there's a lot of complexities with that as well, but we won't really understand the cause of that poor prognosis until we can understand the neurobiology of it.
Oh, that is such an interesting point about the positive correlation in brain size versus, with looking at the brains of autistic individuals. So
Mhm.
really, really fascinating.
Thank you.
Okay, I think maybe just in the interest of time, I know I said we had a healthy amount of time, but I'm realizing we're actually a little tighter. I'm going to just pivot to some of the brain donation specifics now, just because that is the bulk of what sort of came in towards the end here.
Mhm.
So a lot of just sort of clarifying questions. And so I think I caught this one piece on one of the slides, but would just like to know, how does brain donation or does brain donation interfere with funeral services?
Go ahead, Carolyn.
Sure, it's an important and a really good question. You know, I think families are concerned that if they choose for their person to be a brain donor, that they can't carry out regular funeral services that have they may want, so including a viewing. And the fact is that brain donation does not at all preclude a viewing or any funeral services that a family would like to carry out.
The other thing, Dominique, too, what Carolyn has said is true. And when we first started Autism BrainNet, we looked into the major religions of the world to see whether there was any religious restriction on making a brain donation. Um, and we didn't find any. There's cultural differences about how you feel about making a donation, even of other body parts. But of the major religions in the world, there is no. In fact, it's just the opposite. I think most religions would endorse the idea of making contributions to research that might enhance the life of living people in the future.
Great, thank you. That was actually my next follow-up question. So I appreciate you kind of seeing where that conversation was going. I think that's probably something that is on the mind of folks considering brain donation. So thank you. And then in the beginning of the presentation, we looked at the graph comparing the age of individuals whose brains have been donated. So looking at that graph, it looks like you're looking for brains from across the lifespan. Are there specific age preferences at this point in time, or really just?
For just to reinforce the idea, for our donors with the diagnosis, whether that be an autism diagnosis or a diagnosis of a rare genetic condition or both, any age, any sort of circumstances are don't interfere with that person's ability to be a donor to our program. I will sort of circle back to something that David pointed out, that timing is really important. And so even for our donors with the diagnosis, we try to organize the donation process within a really short amount of time, so 24 hours. If time extends well beyond that, we have to think about our ability to honor the family's wishes, because as you can imagine, tissue deteriorates and research is harder and harder. For families that donate for research, we want to be able to honor that to the best of our ability, and we're going to take timing into consideration as we have to. And then for our control donors, or typically developing donors, at this point, primarily we're looking for donors under the age of 50. There are some exceptions to that, but primarily under the age of 50.
I just add to that this is a sort of so first of all, obviously there's not a selection process here other than who will make the decision to be a donor. But I think we have evolved our considerations over the 10 years that we've been going. And now I think while we were interested in younger individuals initially, I think we're now moving to think that it's really important to receive donations from older individuals with autism. And there's some very intriguing, although I think very early data showing that, showing that if you have a diagnosis of autism, it actually increases the chance that you may end up with Alzheimer's disease. This is really, really very new data, and it's going to have to continually be replicated. But and a lot of it is from MRI and behavioral findings. But it would be, I think, enormously important to understand whether at least some subsets of individuals with autism are predisposed to a diagnosis of Alzheimer's later on. The same is true for Parkinson's disease. People like Joe Piven and others have demonstrated now for some time that autistic individuals tend to have a higher incidence of a diagnosis of Parkinson's later on. So again, given that the hallmark features of those diseases are signs in the brain, we would want to see, if there are typical signs of Alzheimer's, for example, in autistic brains at a higher prevalence than in typically developing individuals. So it was really a good question, but I do think Carolyn's notion that, we accept all ages, and, there is important science to be done with donors at each of the different ages.
Across the lifespan.
Great, thank you. And just one last question I want to squeak in here, just for the researchers that joined. I understand some folks might need to hop off. Can you just explain, just reiterate? So in terms of researchers interested in accessing the data and Safari base, is that something that is available to any researchers, or just from those few sites that you pointed out in the beginning of the presentation?
That's an important point of clarification. Any PI is able to apply for tissue or clinical data or clinical data through Safari Base. And we, in fact, David really should speak to this, just that we're distributing tissue worldwide to researchers. If you want to elaborate, David, at all.
Yeah. No, just to say that, any PI can get onto Safari Base, look at what's available, and then put in a formal request. The formal request is evaluated by a scientific review committee just to make sure that the request makes sense. But the Simons Foundation wants this resource to be valuable to researchers worldwide to do state-of-the-art research. So yeah, anybody who, and if, again, as Carolyn said, if investigators have a difficult time accessing the information, please contact us and we'll try and facilitate getting whatever information they need.